to give you an overview of this. So in addition to what is already on the handout, I recommend drawing this out. So if you want to pause the video and then just go ahead and draw your different cells, then that will help you follow along a little bit. So one thing to consider with meiosis is that there are a lot of things about meiosis that are similar in mitosis. So you have the same cellular components that are going to be involved. You still have the chromosomes, you still have the microtubules, you still have the centrosomes and the kinetochores, and all the cellular machinery that is involved in cell division and splitting the cell in two during mitosis is all still here in meiosis. And one of the things that also needs to happen during meiosis is that you also need to duplicate the chromosomes. So just in general, this is a very simplified cell, and it just has one chromosome pair. And in this one, you always receive one copy of your chromosome pairs from your father and one copy from your mother. I did note on here is that just as to orient you in terms of, DNA coding for specific genes, which then lead to specific proteins, then an allele is just a specific version of a gene. So let's say you inherited a brown eye allele from your father and a blue eye allele from your mother. And then depending on the dominance of those particular genes, you get a specific phenotype, which we'll talk about later in the course. But just considering that the chromosomes are coding for specific proteins, and a gene is what's going to code for a specific protein, and allele is just a different version of that gene. So when you have your maternal and paternal copy of a specific chromosome, that is called a homologous pair. So in this particular cell, there's only one homologous pair, right? So there are two chromosomes, and they are a pair. So you received one from the father and one from the mother. In humans, we have 23 homologous pairs. So we have 46 chromosomes total. You received 23 from your father, and you received 23 from the mother. So a homologous pair, right, is just the code, coding for the same genes, and one of them you got from your mom and one you got from your dad. So when a cell is going to undergo meiosis, it does similar to what happened during mitosis. If you are going to make a copy of that cell, then you need to make a copy of the DNA. So the S phase, chromosome duplication or replication, happens in meiosis as well. And so you make a duplicate of the chromosome, and then that gives you the sister chromatid. And in meiosis, it is going to differ from mitosis in one main way. When I ask you what is the most important part of meiosis, it is what happens in meiosis 1 is separation of the homologous chromosomes. So when you've made that copy of the DNA, instead of now lining up at the metaphase plate like it would be in mitosis, so if you're looking at the same cell undergoing mitosis as opposed to meiosis, right? In mitosis, right, we had the chromosomes duplicate, and then the chromosomes lined up at the metaphase plate. The sister chromatids, right, they lined up, and then you just separated the copies that you made. And that was all that happened in mitosis. In order to get an exact copy of the original cell, you make a copy of the DNA, and then you separate those sister chromatids, or you separate the copies. In meiosis, instead of just separating the copies, you first separate the homologous pairs. So now, when the mitotic cell, the meiotic cell, the cell that's undergoing meiosis, now when it goes through that second separation, that's when it separates the sister chromatids. So in meiosis, you have an extra cell division, And that is why you go from a diploid or a 2N cell to haploid gametes, which are the sperm or the egg. And so the haploid is 1N, it has half the number of chromosomes. In mitosis, right, you start out at 2N, you make a copy, but then you split those copies so you end up at 2N at the end. But in meiosis, because you split twice, you go through 2 sets of cell division, you are cutting the number of chromosomes in half. And that is because now you can have sexual reproduction, or a sperm with a 1N or a haploid number of chromosomes is going to fertilize an egg with a haploid or 1N set of chromosomes. And now you take that 1 plus the 1, and now you have a 2N or a diploid set of chromosomes that are necessary in order to make a human or an organism. So it is really important for these cells to undergo these 2 sets of cell divisions so that the gametes will only have half the number of chromosomes necessary to create an organism. So during that sexual reproduction, you're able to have a more diverse set of genes because you're combining the genes or the chromosomes from 2 individuals. So with that knowledge, I'd like you to process that a little bit further. And what I'd like you to do is look at this diagram, and then I want you to create a list of things that are the same for meiosis and mitosis. So kind of create that Venn diagram where you would have things that are involved in both mitosis and meiosis, and then things that are unique to mitosis and things that are unique to meiosis as well. And one of the things you can do is just look at this diagram, right? You know, one thing to start out with is that both of the chromosomes are, both for mitosis and meiosis, you have to have the duplication of the chromosomes. And then if you can, based on what we just previously talked about, and then also looking at this diagram, what are the things that are involved for both mitosis and meiosis, and then individually for mitosis and individually for meiosis. If you can come up with a list, what I will do in the next frame is I'll give you some prompts to be able to put them somewhere, but I want you to think about it on your own. How many can you come up with on your own before I give you the prompts to put them where they are? So go ahead and pause your video and just create a list, and then we'll see how much you are able to get. Okay, so hopefully you were able to come up with at least a few of these. And then, so now that you see all of the different things that are involved, what I'd like you to do is we'll put, so we had already mentioned that there is chromosome duplication for both. So then I would just put, right, DNA synthesis is the same as chromosome duplication. Another term that we're going to see this in coming up in the later chapters is DNA replication. So DNA replication, chromosome duplication, and DNA synthesis are all the same thing. So if you will pause your video, put all the ones that are same down the middle, and then put all the ones that are just for meiosis down over here, and the ones that are just for mitosis over here. So let's continue with the things that are the same, right? So there was DNA synthesis. When you create that, when you undergo that DNA synthesis, what you get are the sister chromatids. So what you had over here was one chromosome, and then when you made that copy, now you have the 2 copies together. So sister chromatids are the 2 copies held together. And then the overall cell machinery is the same. So if you look over here and think about what the cell machinery is that is going to lead to that cell division, you need the microtubules to attach to the chromosomes. The microtubules specifically attach to the kinetochores that are on the chromosomes. And then the thing that organizes the microtubules are the centrosomes. And I think overall that's it for everything that is the same. So now the first process of that DNA synthesis is the same. That created the sister chromatids, and now your microtubules, your kinetochores, and your centrosomes all organize them in similar ways in order to lead to cell division. However, in mitosis, what you can see is that there is only one cell division. So there is only one division right here. In meiosis, there are two divisions. So you have, it's not, oh yeah, it is labeled here. So you have meiosis 1, that's going to be your first division. And then you have meiosis 2, that's going to be your second division. So there are two divisions for meiosis, one division for mitosis. And then because you made the copy and then you divided that once, you end up with the same number of chromosomes as you did for your parent cell. So these have the diploid number of chromosomes. So diploid daughter cells, and if you count how many daughter cells you have, you get 2 daughter cells. In contrast to that, in meiosis, you had those 2 divisions. So you took that one cell and you divided it, and you got 2 cells, and then you took those 2 cells and you divided those again. So now you have 4 cells, so you have 4 daughter cells. And because of that extra division, you had to cut the number of chromosomes in half. So you have the haploid daughter cells. And then it got a little bit small on these ones, but the separation of the homologous pairs happens in meiosis 1. The separation of the sister chromatids happens in meiosis 2. So in mitosis, all you do is separate the sister chromatids and you are done. But in meiosis, the most important part of meiosis happens in meiosis 1, and that is the separation of the homologous chromosomes pairs first before you separate the sister chromatids in meiosis 2. So just to reiterate, without being zoomed in metaphase 1, when they're lining up in the midline, it is the homologous pairs that line up. And then when they separate during anaphase and telophase, it is the homologous pairs that separate. And then during meiosis 2, that is when the sister chromatids are going to separate. And so when these homologous chromosome pairs line up, one thing that you can see happens is that they cross over. So some of the chromosomes are longer and some of them are shorter, and they've actually done experiments showing that the ones that are longer, because the ends of their chromosomes are more floppy, they actually have more places for crossing over to occur. So when this crossing over occurs, what happens is that you get these recombinant chromosomes that are a combination of the maternal and the paternal genes. So crossing over happens in almost all of the chromosomes. So it's not just, oh, I got this chromosome from my father. It is they crossed over when they were lining up in meiosis 1, and then now your chromosomes are really a combination of your maternal and paternal genes. And because only the sister chromatids line up in mitosis, right? That doesn't happen in mitosis. It only happens in meiosis. And then the cells that these are going to create, the mitosis is just going to create your body cells. So if you need more blood cells, if you need more skin cells, or if you need more bone marrow cells, that's just going to make a copy. But if you are going to undergo sexual reproduction you have to cut the number of chromosomes in half so that they can then combine with the DNA of a different individual for sexual reproduction. And that is the formation of the gametes, which happens in meiosis.